Good morning, dear children. Standard 11th, I again welcome you all with the same chapter, Systematics of Living Organisms. Yesterday, we saw in this chapter about classification, the criteria is used for classification. We saw artificial method of classification, natural method of classification, and phylogenetic method of classification. Artificial method, which was used by Linnaeus, in which he used only the things which are observable. In natural, we saw they used significant things. And in phylogenetic, we saw evolution was the main criteria to study it. Phylogenetic method of classification is used by Bentham and Hooker, by R.H. Whitaker and Carl Woese in their studies, and we are implying it and using it. Phylogenetic method of classification is a wonderful method of classification because it speaks very well about the evolution, right from a single prokaryotic cell to a multicellular cell. So the difference, what we get to see in this, is very well observed in this phylogenetic method of classification. To begin with today, we are going to talk about first the taxonomic categories. After that, we'll talk about taxonomic hierarchy. And after that, showing you the hierarchy, we will begin with the five kingdom system of classification, which I have got for you all in the PPT. So yeah, okay. So here we can see we are again talking about the same topic, systematics of living organisms. Taxonomic categories. What is taxonomic category? Classification is not a single step process but involves hierarchy of steps in which each step represents a rank or category. It is just like in school you must have seen there are ranks. First is the principal, then the vice principal, then the supervisor, then the teachers, and even for you must have seen head boy, head girl, assistant head boy, assistant head girl. So are the ranks, and in the same way in classification we have ranks. We have proper step by step broadness brought in it, and then we arrange it accordingly. We arrange the living organisms accordingly. This is what is called taxonomic categories. Classification is not a single step process but involves hierarchy of steps in which each step represents a rank or category. Since the category is a part of overall taxonomic arrangement, it is called taxonomic category, and all categories together constitute the taxonomic hierarchy. Kingdom, division, class, order, family, genus, species are the categories in hierarchical sequence. These are compulsory categories. Besides, there are some facultative categories like suborder, subfamily, etc. to be used as per the need. Okay. So this is taxonomic category. Now this is taxonomic hierarchy in the way we classify a thing. Here we have taken two classifications. One classification is of hibiscus rosa sinuses. जास्वंद जी लाल रंगाची आहे त्याचं एक क्लासिफिकेशन आपण घेतलेलं आहे आणि ह्याच्यामध्ये आपण दुसरं क्लासिफिकेशन जे घेतलेलं आहे ते आहे कोब्राचं, कोब्रा स्नेकचं. त्याला हिबिस्कसला चायना रोज सुद्धा म्हणतात, त्यामुळं लिहिताना आपण लिहिले क्लासिफिकेशन ऑफ चायना रोज. दॅट इज अ वन आय कॅन से अ नेम विच इज व्हेरी कॉमन टू अस, वर्नाक्युलर और लोकल नेम चायना रोज. Now over here if you see properly, category and taxon, both the columns are shown. Kingdom Plantae has a division Angiosperm, in which the class Dicotyledone shows a subclass Polypetalae and has a series called Thalamophlori with the order Malvaceae, has a family Malvaceae, has a genus Hibiscus, and the species is Rosa sinuses. This I came from upside down. Now actually it goes from below, from the basic to the uppermost part. We get a flower. We don't know what the flower is. I have got a flower. Today I know this is called an hibiscus flower. But previously when people searched it, when they never knew about it. They got the flower, they saw the flower. Okay, and after that, it was placed in the kingdom plant because it came from the plant. Which kind of plant? Angiospermic plant. Then the plant has dicotyledon class. How come we have given it a dicotyledon class? There are two classes, monocotyledon and dicotyledon. What is the specification of a dicotyledon and a monocotyledon? You will tell me that. Polypetally, many petals. When there are two petals, bipetally. Monopetalous, bipetallous, tripetalous, tetrapetalous, and polypetalous means more than this. So basically, this plant or this flower has 5 petals, polypetally. Thalamophory. Everything is arranged on the thalamus properly. Malveals is the order. Malvaceae is the family. Genus is Hibiscus and species is Rosa sinensis. This is the way the hierarchy goes on. Now, this in this way only the unit of classifications are given in your textbook. I would just like to go through it, read it, so that you understand what exactly it is. Species. Species is the principal natural taxonomic unit ranking below a genus. Where is it present? Species below the genus, denoted by Latin binomial, considered as the basic unit of classification. What is it? It is the basic, the very first step of classification. It is a group of organisms that can interbreed. These varieties of hibiscus can interbreed with each other. There are several varieties. They can interbreed with each other under natural condition to produce fertile offsprings. It was thought to be an indivisible, stable and static unit. However, in modern taxonomy, subdivision of species such as subspecies, varieties and population are seen and given more importance. Now, genus. Genus is a taxonomic rank or category larger than species. Second step or one step above species is genus. It is larger in size. I told you the vastness goes on increasing from the lower level to the higher level. Used in biological classification of living and fossil organisms. Genus is a group of species bearing close resemblance to one another in their morphological characters, but they do not interpret. This information is given in your textbook. I have always been telling you, refer your textbook, read it. Though the syllabus is deleted, please go through it. See, now here you will understand what exactly a species is and what is a genus. Now we are talking about genus. Genus is a group of species bearing close resemblance to one another in their morphological characters. There is resemblance in what? In the morphological characters, but they do not interpret. There are many members in the family Malvaceae, but all cannot breed with each other. They have some similarities, some differences, and that's why they are placed there. Talking about genus, here they have given a very good example over here. Example: tiger, leopard, lion. All three belong to the same genus Panthera, but they cannot interbreed with each other. They have common characters yet are different from each other because their genus is same but the species is different. Another example of genus is Solanum. Brinjal and potato both belong to the family Solanaceae, Solanum. But they cannot interbreed with each other. Family. It is one of the major hierarchical taxonomic rank. A family represents a group of closely related genera. Example genera like hibiscus, gossypium, syra, bombax are included in same family Malvaceae. Cat also belongs to family of leopards, tigers and lion family. Felidae, but dog belong to different family. Canidae. Canidae carnivorous or it is a part of carnivorous only actually, but a different family. Hmm? Cohort or order. It is a taxonomic rank used in the classification of organism and recognized by nomenclature codes. An order in a group of closely related families showing different affinities. Order thus is a step above the family. First we saw species, then came genus, then came family, then came order, then came the class. Class is the distinct taxonomic rank of biological classification, having its own distinctive name. A group of higher taxonomic rank than order. Class is the assemblage of closely allied orders. Order Carnivora and order Primates belong to the class Mammalia. Thus, monkey, gorillas, gibbons, primates, and dogs, cats, tigers, carnivore belong to the same class. Division or phylum, subkingdom, and kingdom are the next steps involved in this. Division, a category composed of related classes, example division angiospermy. Angiospermy has a vast variety of plants. Today we get to see they are the dominant ones. Hmm? So it becomes more vast as compared to species, genus, family, order, class, and division. Now, division. The division is a category composed of related classes. Example division angiospermy includes two classes, dicotyledon and monocotyledon. It is based on the cotyledon, as well as how do we say that this plant is an angiosperm? A flowering plant, seed bearing plant, seeds are enclosed in the ovary. When the seeds are enclosed inside the ovary, we place them in the division angiospermy. Why do we do this? This is a characteristic feature of this division. And the most important thing, an example of this, you eat mangoes, they are enclosed in an ovary. You eat other fruits, like, see, there is a difference between a true fruit and a false fruit. If you look at apple, apple has a very small covering. It's not a true fruit, it's a false fruit, because the thalamus over there goes on increasing. This we will be studying in the coming lectures. I just want to make you understand what exactly, what is the criteria of placing a plant in angiosperm. When I say angiosperm, the seeds are present. Seeds are enclosed in the ovary. They have a covering over there. They are not naked. They are seed-bearing, flower-bearing plants. Okay, so this is the characteristic feature of angiosperm, of a division. Now, when I say a division, it includes two classes, dicotyledon and monocotyledon. Monocotyledons also bear seeds, but the difference is they bear only a single cotyledon. And here there are two cotyledons in a seed. That is the basic difference. Subkingdom. Different division having some similarities from subkingdom. Example, the division angiosperm and gymnosperm will the subkingdom phanerogams and spermatophyta. Phanerogams and spermatophyta are the two subdivisions which we get to see in the kingdom Plantae. Kingdom. It is the highest taxonomic category composed of different subkingdoms. Example, subkingdom Phanerogams, Cryptogams. Now, looking at all this, kingdom Plantae, in just a short view, I would like to tell you. Kingdom Plantae is divided into two important divisions. Phanerogams cryptogams. Phanerogams seed bearing, cryptogams spore bearing, which are very small. This is the basic difference between them. Phanerogams are further classified into two different divisions. One is angiosperms and gymnosperm. Okay, this is how kingdom Plantae is classified. Angiosperm, gymnosperm. Angiosperms are further classified into class dicotyledony and monocotyledon. So kingdom नंतर काय येतं? What is there after kingdom? Subkingdom, division, and this is the class. I hope you understood this. Did you all see this? Kingdom Plantae is divided into the two sub-kingdoms, phanerogams or cryptogams. Cryptogams are also called as spermatozoans. They are further divided into the division angiospermy and gymnospermy, class dicotyledon and monocotyledon. And this is how the hierarchy goes on. I hope you all have seen it, can see it well. If not, in the next lecture, again, we will go through it. Any difficulties, you can contact me. No issues about that. Nomenclature. Any object that becomes known to human intelligence must possess a name, and it may not be possible or convenient to describe it in order to communicate ideas about it. The art of naming the object is in fact a science called nomenclature. When we find something, when the human found something, it always named it something. Always a name was given, and this naming process is nothing but called nomenclature. Now there are three types which we are going to see. Two basic are there, and then we will see what is the important one. One is a vernacular name and the other is a scientific name. Vernacular name is a local name which we use in our areas, while a scientific name is a name which is used by everybody, especially the scientists in the world. Wherever you go and you say, can I get Azadirachta indica, they will give you neem plant immediately. If you go and say in US, I want Mangifera indica, they will give you a mango immediately. But if you go there and say, can I get a mango, don't know in China whether they call it mango or what else. In Japan, if you go and say, can I get neem plant, I don't know what you will get the answer. The reason is this, that this knowledge which is gained by us shouldn't be restricted. It should be open for everyone, and therefore came into existence the binomial nomenclature. Binomial nomenclature is nothing but a system of naming. How to name it? Now, how to name it? Name of organism is composed of two Latin or Greek words. Generic name is simple noun. It should come first and begin with a capital letter. Specific name is the descriptive adjective, which should come later and begin with a small letter. Scientific name must be underlined separately in handwritten and must be printed in italics. The generic and specific name. Now, over here you can see the genus and species, both are written in a different font. On the PPT, you can see that very well. This is how we write the binomial nomenclature. Name, scientific name, Hibiscus rosa sinensis. Hibiscus is coming in capital and rosa sinensis is coming in small, but both are underlined. What is done? Both are underlined. Scientific name must be underlined separately if handwritten and must be printed in italics. You will see this is written in italics on the PPT. The generic and specific name should not have less than 3 letters and more than 13 letters. Usually the name of the author who named a plant or animal is also written in full or abbreviated form after scientific name is given. For example, Mangifera indica. Mangifera indica is the name of, I don't think so, there is a duster over here. Scientific name of mango is Mangifera indica. Indica. And with that, they always give an abbreviation over there, that is L dot. Mangifera indica L. Why this L is given, this abbreviation is given? Because this name was found by Linnaeus. In respect to him, this L is abbreviated over here. Indica, it is found in India, that's why Indica. Mangifera, a genus of mango. This is how the scientific name is written. Now you have understood what is classification, you have understood what is nomenclature, what is taxonomy, and without that, how would I explain you all the five kingdom system of classification? There were criteria, there were names given, and all this has some rule, some I can say some rules to follow, and that's why I had to explain you these rules so that you can understand the five kingdom system of classification. Let's see our next slide. It was R.H. Whitaker who classified all the living organisms using phylogenetic relationship among them. We are talking about the classification now, and this is what is the part which is not deleted from your syllabus. Previous what I taught was deleted from your syllabus. Now this is what is there for your exam. So please go through it. Okay. Five kingdom system of classification by R.H. Whitaker. R.H. Whitaker has classified all the living organisms using phylogenetic relationship among them. He used the method of evolution over here. Carl Linnaeus also classified the living organisms. In your textbook, there is a short information given which we won't omit, we will definitely read it. Carl Linnaeus classified living organisms in two kingdoms based on mode of nutrition, whether they are migratory, sedentary, etc. But this broad classification as kingdom Plantae and kingdom Animalia was found inadequate. Though Carl Linnaeus classified them, but still it was inadequate. The information was less. And to keeping in mind this, it could not classify the organisms that show character of both the kingdoms. Example, bacteria, fungi, and euglena, etc. Hence, to avoid the confusion, scientist R.H. Whitaker in 1969 proposed the five kingdom system of classification. The system shows phylogenetic relationship between organisms. Five kingdom system of classification shows these five kingdoms: kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. Now, if you see, I have written the three first and the two down. There is a reason for it. I could not place an arrow actually in this. I'm very sorry for that. I'm very bad at using computer actually. But telling you very frankly, kingdom Monera, the name itself, mono era, an era where the organisms were present which were unicellular. How were they? Unicellular, primitive ones. Mono, single, era, beginning. This was the beginning. And that's why we can see a major difference. We can observe one thing, that only the unicellular prokaryotic organisms are placed in kingdom Monera. Then we come to kingdom Protista, where a little bit transformation is taking. The prokaryotic cells are moving towards eukaryote. In Monera, if we talk about the three domain theory, we saw archibacteria and bacteria. Both are placed in kingdom Monera. But when we speak about kingdom Protista, then comes the eukarya. Eukarybacteria, in which the eukaryotic cells came into existence. Here we see a vast variety, a vast difference occurring in the kingdom Protista, from unicellular to multicellular, eukaryotic, from prokaryotic to eukaryotic. So Protista was a bridge. Somewhere it was a bridge for kingdom Monera and kingdom Fungi. It was just a link between these two. From Protista we go to kingdom Fungi, which also shows a bifurcation from unicellular to multicellular, from spore to filamentous body, fruiting bodies, and then the bifurcation goes on. The variety is extending, it's changing. Eukaryotic cells are seen. We see and we observe these changes over here, and from there, from this fruiting bodies and spore formations, we go on to a new kingdom called kingdom Plantae, where the chlorophyll pigment is present. Heterotrophic mode of nutrition can be seen, and a variety can be seen over there also. And then comes kingdom Animalia, where locomotion can be seen in the organisms. We can see that they are autotrophs depending on others. Some are carnivorous, some are herbivorous. The vast differences are just nothing but links between each other. That's why kingdom Monera is the beginning, Mono era. Protista, where the protest began. Protesting, you are going against, or you are needing of a change. We have always seen changes have occurred when people have protested. It's the same in protesta. The word itself gives an idea. I love these words. The reason is these words always give us an idea about what exactly must have happened over here. Even the organisms protested against what exactly was in the beginning, and now they are going to a change in the kingdom Protista. Then comes kingdom Fungi, then comes kingdom Plantae, and then comes kingdom Animalia. Today we will be talking about kingdom Monera. These points which are given in the PPT are very important. Read them. They are from your textbook. I have not got any other grad material from anywhere. It is very well defined in the textbook, and that's why I have used the same language. No change in the language. Unicellular organisms. How are the organisms in Kingdom Monera? They are unicellular, single cell. Prokaryotic cell organization. Prokaryotic primitive nucleus. Primitive. Karyos nucleus. Cell organization is there. They are omnipresent. They are present everywhere. Omnipresent means present everywhere. The meaning of omnipresent means present everywhere. They are photoautotrophs and chemoautotrophs. What are they? Photoautotrophs or chemoautotrophs. Photoautotrophs using light. Chemoautotrophs, chemicals. Majority are heterotrophic in nature. These organisms do not have a well-defined nucleus. As we know, we use the word prokaryotic, means they do not have a well-defined nucleus in them. The DNA exists as a single double-stranded circular single chromosome called nucleoid. DNA is present. Now, let me tell you one thing which I told you yesterday also. Primitive or prokaryotic cell do not have a well-defined nucleus. We are going to come with this statement again. But let me insist on this once and for all, again and again. When I say it is a prokaryotic cell, means the nucleus is not well-defined. It is not well defined means it does not have a nuclear membrane, it does not have a nucleoplasma, it does not have a nucleolus, and because it does not have all this, we cannot say that the DNA is concentrated inside the nucleus. No, over here the DNA is there. Every cell, every organism has a DNA. Even organisms of kingdom Monera have a DNA. But how is the DNA? It is not in the nucleus. It is existing as a simple double-stranded circular single chromosome called nucleoid. It is not present in the nucleus. Somewhere in the cell, it is present as a simple double-stranded circular single chromosome. Now, the cell walls are made up of peptidoglycan, which is a polymer of sugar and amino acids. Membrane-bound organelles are absent. Membrane-bound organelles like endoplasmic reticulum and all the other ones like chloroplast, mitochondria, these are all absent in this kingdom, Monera. We said that it is a primitive cell. What is it? A primitive cell. When we say it is primitive, that means it did not have a well-developed body in it, and it's a unicellular organism. Okay. Now after that, ribosomes are present, but they are smaller in size of 70s than in the eukaryotic cell. In eukaryotic cell, the ribosomes are much more bigger. Mode of reproduction is asexual or with the help of binary fission or budding. Very rarely sexual reproduction is by conjugation method. How is the mode of reproduction? Asexual. Asexual, where both the sex organs are not involved. The single parent is involved in it. That reproduction is called as asexual. You have studied in your 10th about amoeba. You have studied about yeast. You have studied about Spirogyra. These all are a kind of uh, reproductions which we are talking about over here. Morphologically, bacteria are categorized in four groups. Now, Monera shows bacteria because it has bacteria present in this kingdom, which are present everywhere. The second statement or the second salient feature we spoke about kingdom Monera is that they are omnipresent, present everywhere. Bacteria are present everywhere. Now, what are the bacteria type? Categorized on spherical type, Coccus, rod-shaped, Bacillus, comma or kidney-shaped, Vibrio, and spiral, Spirulum. These are the different types of bacteria which we get to see. This is all about the salient features of kingdom Monera. I want you to write these salient features in your book very properly. Now, on the basis of evolution, on the basis of evolution, bacteria can be classified as archibacteria and eubacteria. Archbacteria. I'll explain this part. I have this is a part which includes in kingdom Monera. Once we finish explaining this part, kingdom Monera will be over. But don't just leave it. Kingdom Monera is very easy to understand. Write down the salient features in a book. This question will definitely come. Write the salient features of any one kingdom out of the five kingdom system, or mention the five kingdom system of classification. This is the favorite question of this topic, and only this part is there. That's why you go through it. Okay. We will talk about archbacteria. Bacteria are present in kingdom Monera, and we are talking about archbacteria. These are different from other bacteria on the basis of their different cellular features. These bacteria are mostly found in the extreme environments, hence termed as extremophiles. Where are they found? In the extreme environment. What do you mean by extreme environment? Very cold or very hot. That's why they are called as extremophiles. They are found in variety of places, from volcanic craters to salty lakes and hot springs. I told you, एक तर खूप उष्ण नाही तर खूप थंड अशा दोन्ही ठिकाणी असल्यामुळे त्यांना काय म्हटले? एक्स्ट्रीमोफाइल्स. Their ability to withstand such hostile environments speaks of their capacity to survive in very severe conditions. Bacteria survive at very high temperatures also, and that is what is mentioned over here. Bacteria that can withstand high salinities are called as halophiles. खारटपणामध्ये येणारे काही बॅक्टेरिया आहेत, खाऱ्या याच्यामध्ये, खाऱ्या पाण्यामध्ये सुद्धा, त्यांना काय म्हटलं जातं? हॅलोफाइल्स. दे आर कॉल्ड एज हॅलोफाइल्स. While those withstand the extreme temperature are known as thermophiles. A very common example is the methanogenic bacteria found in gut of ruminant, that is cows and buffaloes. त्यांच्या पोटामध्ये जो बॅक्टेरिया सापडला जातो तो एक एक्झाम्पल आहे. These bacteria help in the production of methane in biogas production. Biogas production मध्ये कशाचा उपयोग होतो? Those bacteria which come out from the cow dung, from the stomach or the intestine of cows and buffaloes. Eubacteria. This is the last part. These are commonly referred as true bacteria. They have cell wall of peptidoglycan. They are found as autotrophs or heterotrophs. The autotrophs can be photosynthetic like chlorobium, green sulfur bacteria, and chromatium as chemosynthetic like sulfur bacteria. These are mostly multicellular filamentous forms living in fresh water. The body is covered by mucilaginous sheath. The genetic material is typical prokaryotic, that is chlorophyll A and chlorophyll B. Carotenes and xanthophylls are the photosynthetic pigment. These are the photosynthetic pigments present in the plant. Underlying this statement, presence of these chemicals or these pigments is nothing but an indication that they are autotrophic. Filaments show heterocysts which help in nitrogen fixation. Heterotrophs are most abundant. Most of them are decomposers and known for breaking down large molecules in simple molecules and minerals. They can be anaerobes, helping in curdling of milk, that is lactobacilli. दुधापासून दही तयार होणं. Lactobacillus is a bacteria which is used. Fixation of nitrogen. Azotobacter. मेथीच्या नोड्यूल्स मध्ये असतात, रायझोबियम असतात. Antibiotic production, streptomycins. Composting and degrading oil. But the story doesn't end here. Some of them are pathogens causing diseases: typhoid, cholera, tuberculosis, tetanus, etc. Mycoplasma, these are smallest of the living forms. They do not have cell wall. Many forms are pathogenic. They are found resistant to antibiotics due to absence of cell wall. Now, you will say, I have been still showing the PPT and reading from the textbook. The thing is, this is totally a part of kingdom Monera. Now, we are done with this PPTs. I will just talk to you about kingdom Monera once again in detail. What did we see in kingdom Monera? These are unicellular prokaryotic organisms, omnipresent. How is the mode of nutrition? They are either photoautotrophs or chemoautotrophs, using light or chemicals. Peptidoglycan is present in the cell wall. After that, how is the mode of, they are heterotrophic, dependent. As well as we saw the mode of reproduction, asexual and sexual, both are there. But sexual is in the type of conjugation. Asexual, budding, binary fission. These are the methods of reproduction in them. Now enlist all these salient features. Write them down in your notebook. The notes which you are going to make, point wise, write it over there. Five kingdom system of classification by R.H. Whitaker. R.H. Whitaker classified living organisms into five kingdoms. The five kingdoms are kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia. Kingdom Monera. And then write down the salient features which I have shown in the PPT. See the lecture twice, thrice. All the points are there. They are there in your textbook also. We spoke about Archbacteria, Eubacteria and Mycoplasma also. These are all the bacteria which are involved in kingdom Monera. Because this is an kingdom which has such small organisms which are microscopic, unicellular and present everywhere. With this, I have done with the kingdom Monera. I hope you all have understood it. If not, any queries, let me know and contact me as soon as possible. Write down all the notes which I have given you and continue writing it and learning it, because there will be a definite question on kingdom, five kingdom system of classification. God bless you all. Have a wonderful day.